Hello and welcome to my channel. My name is Shisha, and in this particular lecture we are going to be talking about flow cytometry. Now some of you have already left comments that this is a topic that you would like me to cover. So here we are. We are going to discuss about flow cytometry in this particular lecture. So what is flow cytometry? Well, we will be discussing about it in details, but let it suffice to say that it works on the principle of immunofluorescence. So a couple of lectures back, we talked about immunofluorescence, and one of the applications of immunofluorescence was flow cytometry. So we also talked about the other applications of immunofluorescence, where we talked about immunohistochemistry and immunocytochemistry. So those IHC and ICC are used for visualizing tissue sections and cells respectively. In flow cytometry, the detection works more at the cellular level. So as we go through this lecture, it will become clearer. It is I wouldn't say it is very common, but it is pretty commonly utilized in different laboratories for sorting cells or for counting cells in a population to identify perhaps the differential distribution of different cell populations in any specific system. So without any more delay, let us just dive right into the topic of flow cytometry. So flow cytometry is a technique for counting, examining, and sorting microscopic particles suspended in a stream of fluid. Now this right here is a very important point to keep in mind, that these microscopic particles with which all of these things are going to be performed, they have to be suspended in a stream of fluid. So what you require is actually a suspension of cells. If the cells are clumped together or aggregated together, then that will not really work. So a suspension of cells is the first and foremost requirement for performing flow cytometry. So flow cytometry can help in not only counting and examining the cells, but also in sorting the cells. So the flow cytometer uses a laser beam and a light detector to count single intact cells in suspension. So you can actually count individual cells present in suspension using flow cytometry, and that is how sophisticated this entire system is. It utilizes a laser beam and a light detector. Now, exactly what roles both of these play, we will talk about it in details. So every time a cell passes the path of a laser beam, so light will be deflected from the detector, and this interruption of the laser signal is recorded. So assume that there is a laser, and right in front of the laser you have a detector. So in absence of any cells passing through the path of the laser, the signal of the laser would be detected by the detector in a continuous and uninterrupted fashion. Now, as a cell passes through the path of the laser beam, it will deflect the laser beam. How so? So the laser beam is moving, it will hit the cell on its way, and it will now get scattered from there. So this will lead to deflection of the laser beam, and as the laser beam is deflected, the detector can no longer detect it, because the detector previously was detecting a continuous input of laser beam, and now as the cell passes through the path, it breaks the path of the laser beam, and there is a momentary lapse in detection, and this interruption of the laser signal is what is recorded. Those cells which have a fluorescently tagged antibody bound to their cell surface antigens. So this is where immunofluorescence come in. So you will have specific antibodies coupled to specific fluorophores, and these antibodies would be for specific cell surface proteins. So if a particular cell is expressing this particular cell surface protein against which you have added the antibodies, so then what would happen is that this fluorophore, which is attached to the antibody, would be excited by the laser, and once excited, it will emit this light, that is recorded by a second detector system located at a right angle to the laser beam. So there are now two detectors that we have talked about so far. The first detector was detecting an uninterrupted flow of laser. So it was bam right in front of the laser beam. As the cell is passing through, it is breaking the flow of the laser or the path of the laser, and this detector is not receiving any signal momentarily. So this lack of signal by detector 1 is the first thing that is recorded. Next, if this cell has or is expressing the specific epitope against which you've added an antibody, so this antibody is conjugated to a fluorophore. The fluorophore will be excited by the laser, because keep in mind that the laser beam would be of such a wavelength that can excite the fluorophore molecule. So as the fluorophore molecule is excited, it will now come back to its ground state, and in doing so, it will emit light of a longer wavelength, and this emitted light will now be detected by the second detector system. And this second detector system is located at right angles to the laser beam. Why? Because under such placement conditions, how do I put it? So the laser beam is obviously moving in a straight direction, but some waves of light will deflect or will not really be moving in a straight line, like directly from the laser to the detector. Okay, wait, this will make it clearer. So let us say you have this laser beam over here and it is coming out. This is the source, this is a laser beam, and this here on the right side is the detector. Now, some of these beams might be going like this. So if the detector is at a right angle to the laser beam, like somewhere over here, then the chances that these deviant rays will be detected by this detector is lower. So this placement helps in minimizing the noise or the background to say. Okay. So the simplest form of this instrument counts each cell as it passes the laser beam and records the level of fluorescence that the cell emits. So how is it counting each cell? So the first detector, which is detecting the continuous flow of laser, so that will count the cell, because irrespective of whether the cell has this specific fluorophore conjugated antibody associated with its surface, the pathway of the laser would be interrupted as the cell passes through. So this lack of signal will be counted, and this will give us a count of the total number of cells. And within that, those cells which express the specific protein against which you're adding the antibody, they will in addition to this also emit fluorescence, and this emitted fluorescence would be detected by the second detector. So the first detector is counting the total number of cells. The second detector is giving you an idea about the number of cells that are expressing this particular protein against which you have added the antibody. So this will actually allow you to estimate the fraction of your total cells which are expressing the specific protein. So the simplest or the most basic form of a flow cytometer just counts each cell as it passes through, as well as records the level of fluorescence that the cell emits. Now there is an attached computer that will generate a plot of the number of cells as the ordinate and their fluorescence intensity as the abscissa, that is the X versus Y plot of it. Now, more sophisticated versions of this instrument is a fluorescence assorted cell sorter or fluorescence assisted cell sorter or FACS, which is not only capable of performing this function of counting the number of cells, but also capable of sorting the population of cells into different containers according to their fluorescent profile. So it is a advancement of this simple flow cytometry, where not only are you counting the number of cells, but you're also sorting them on the basis of the level of fluorescence. So this is a schematic layout of flow cytometry. So you have, see, the excitation laser over here, and this is the first detector. So you see it is just right in front of the excitation laser. So this is where you are feeding the cells into the system from, and as each and every cell makes its way through, so whenever a cell is passing through, it will deflect the laser beam, and there will be a lack of signal detected by this first detector, the FSC detector, which is called the forward scatter detector. Now, as the light is being deflected, it can be detected at a number of different levels. So these are all dichroic mirrors or optical filters. So dichroic mirror, what they are, we have talked about it. So they are mirrors that allow light of one wavelength to pass through, whereas it will reflect light of other wavelengths. So as you can see over here, like to begin with, here you have four colors. You have blue, green, yellow, and red. So this mirror is allowing only red, green, and yellow to pass through uninterrupted. This filter is allowing red and yellow to pass through uninterrupted. And this one is allowing only the red beam to pass through uninterrupted. So the system is designed such that there are several different filters present, and each of them are positioned in such a fashion that the beam or the specific wavelength of light that can pass through it will then be detected by the specific detector. So the FL is nothing but the fluorescence detector. So we have three different fluorescence detectors, which means that this particular system is capable of detecting fluorescence from three different fluorophores at three different wavelengths. So you can use three different antibodies conjugated to three different fluorophores for the purpose of your cell counting to identify multiple populations of cells. Right. So let me break that down. Let us assume that your population has several cell types, and there is type 1 which expresses only epitope A, type 2 which expresses only B, then there is a type 3 which expresses only C. Now you can also have subtypes which express a combination of these epitopes, like say for instance a type 4 that expresses A and B, a type 5 that expresses A and C. So this system is sophisticated enough to distinguish between or among these individual cell types. Okay, so we have in this particular schematic three different fluorescence detectors. Now this is the SSC detector, which is the side scatter detector, and this essentially measures scatter at a 90-degree angle relative to the laser. So this is roughly 90 degrees. Well, it might not really be clear in this particular schematic, but this is the second detector that we were talking about, which is placed roughly at 90 degrees. Okay, so the system is so designed that only one cell will pass through the pathway of a laser at a time, and that is how it is supposed to work. And because this is the basic system where you're not really sorting the cells, so as the cells pass through, they're going to a waste container. So once the detectors detect the signal will be amplified. There will be an analog to digital conversion, and now the computer will analyze the result and give you an output of how many total cells there were, what proportion of cells were emitting only this green fluorescence, what proportion was emitting only red fluorescence, what proportion was emitting a combination of say yellow and green fluorescence, and so on and so forth. Okay. So this is the main concept that you have to grasp of how the flow cytometer works. So a flow cytometer has five main components. A flow cell. So liquid stream or sheath fluid carries and aligns the cells so that they pass single file through the light beam for sensing. So essentially, in simple terms, this means that the flow cell is such that at a time only a single cell will pass through the path of the laser, and only a single cell will interrupt the laser. So whenever there is a absence of laser signal detected by the first detector, that is the forward scatter detector, the FSC, this means that there is only one cell. So because this lack of signal would be counted as one. So if there are more than one cells, then you can't really say how many cells there were. That is why it is important that you have a uniform cellular suspension. Okay, so that only one cell passes through the path. There is a light source. Commonly used include lamps of mercury or xenon or a laser. A detector and analog to digital conversion system, the ADC. So these detect forward scatter and side scatter as well as fluorescence signals. An amplification system, which can be either linear or logarithmic. And finally, a computer for the analysis of the signals. So the computer will basically compile the information detected by all of the detectors and give you the output. So okay, this is the total number of cells. This is the population of cells that fluoresce this and this. This is the population of cells that fluoresce that and so on and so forth. So this is what atypical flow cytometer looks like. Everything, all of the detectors and everything is present within this big machine, and the flow cell is also inside it. So sometimes, in fact, it becomes important to resuspend the cells as they are placed in the flow cell. So you can't just add your cells to the flow cell and leave it there indefinitely. So you add little by little cells and you resuspend them to ensure that the cells are not really clumping together as they are settling down in the container under the effect of gravity. Because you have to keep in mind that cells will settle down under the effect of gravity if kept static for a prolonged period of time. So that has to be avoided. And also the cells are maintained at a dilution of typically around 1 million per ml. So it is not too concentrated that several cells can together interrupt the pathway of the laser or the flow of the laser. Also, the nozzle, rather, if you go back to the previous slide, that narrow channel through which the cells were passing in front of the laser, the channel is thin enough to ensure that only single cells can pass through and clumps of cells do not make their way through it. So now we are going to talk about FACS, which is an extension of fluorescence activated cell sorting. I'm sorry, which is an extension of flow cytometry. So more sophisticated versions of flow cytometry is fluorescence activated cell sorting or FACS. Now, FACS provides a method for sorting a heterogeneous mixture of biological cells into two or more containers, one cell at a time, based upon the specific light scattering and fluorescent characteristics of each cell. So the sorting is not only based on the fluorescence characteristics, but also on the light scattering properties. And the light scattering properties will be dependent on certain physical parameters like the size of the cell and so on and so forth. So it is a useful scientific instrument as it provides fast, objective and quantitative recording of fluorescence signals from individual cells. So it is very important to keep in mind that whatever information that you're collecting from FACS or from flow cytometry for that matter is of individual cells. And not only does it provide this quantitation, but also allows physical separation of cells of particular interest. So let us say you have a population of cells and you want to isolate only a specific type of cells from that population that is expressing specific markers on its surface. So FACS will allow you to do that. So a common example would be you have blood cells and you want to identify specific type, or you want to isolate, specific type of blood cells that are expressing specific markers on their surface. So the best way to do that is by doing a FACS assay, where you take your blood cells, you prepare the suspension, you let it pass through the machine, and you obviously, prior to letting it pass through the machine, incubate it with antibodies specific for the specific markers, and then this machine will simply sort out this specific population of cells, and you can then do further experiments and further analysis with it. So this is a cytogram of bacterium samples taken from pine oak litter and bulk soil. So you can see how the distribution pattern is varying between these two sample sources. Okay, so exactly what it means or what it is detecting, I'm not really going to discuss with you because that is outside the purview of this lecture. But this is what a typical flow cytometry cytogram looks like. And this red encircled area essentially highlights the cells of interest. So this is referred to as gating. So you have to identify the parameters based on which you will say, okay, so if my cells fall under this particular area, only then are they of interest to me and not otherwise. So this definition or defining this parameter or this area is called gating of the cytogram. So let us talk a little bit more about facts and how exactly it works. So what are the extra steps over here? So the stained cells, that is the cells which have been incubated with these fluorescence conjugated antibodies, are loaded into the sample chamber of the cytometer. So the first step would be that you allow the cells to incubate with these fluorophore conjugated antibodies, and once they have incubated and the antibodies have bound, you wash it to remove of the excess antibodies from the solution, because the excess free antibody molecules in the solution would be interfering with the detection, and then you load the cells into the sample chamber of the cytometer. So the suspension is expelled in the center of a narrow, rapidly flowing stream of liquid, and it is expelled such that at a time only a single cell passes through, because this stream passes through a very fine capillary which is so thin that only a single cell can pass at a time. The flow is arranged so that there is a large separation between the cells relative to the diameter. Now this step is important to allow for the sorting of the cells, because over here you'll be collecting these cells depending upon the pattern of fluorescence or fluorescent signal emitted from them. So this gap between subsequent or successive cells will help with the sorting. Because what if that there are two successive cells and one of them has a different fluorescence pattern and the other one has a completely different fluorescence pattern, and both of them have to be collected in separate containers. So you have to allow some gap in between these individual cells so as to allow the machine to be able to resolve these two successive cells. Okay, so this therefore the suspension should not be very concentrated or confluent, and it should be pretty dilute to allow for this separation. So the cells are expelled one at a time from a small vibrating nozzle that will generate microdroplets. Each microdroplet will contain a single cell. Okay, so it's in a stream of liquid and the stream is expelled from this vibrating nozzle. So as it comes out, it forms these microdroplets, each having a single cell. Now, as it leaves the nozzle, each droplet receives a small electrical charge. So this electrical charge would actually help with the sorting later on. We will discuss how. So as the cells leave the nozzle within a micro droplet, each droplet receives a small electrical charge, and the computer that controls the flow cytometer can detect exactly when a drop is generated by the nozzle, and this drop passes through the beam of laser light that excites the fluorophore. Okay, so the computer system is hooked up such that it can detect exactly when this drop passes through the beam of laser. Okay, so this is a schematic representation of facts. So you have your cell suspension and you're feeding it into the flow cell. And this is and the ultrasonic nozzle vibrator. The vibrator will help in the formation of these droplets. Each droplet has a single cell. Now the moment it comes out from the path or from the flow cell, it is hit by the laser. And these are the two detectors. The first detector, which will count the number of cells, and this is the second detector at a 90 degrees angle to the pathway of the laser, which will detect the fluorescence. So as soon as the cell comes out from the nozzle, it will hit the laser pathway and the detectors will detect the presence of the cell as well as the fluorescence emitted by it. Next, what happens is that these droplets are allotted some charge. So first the laser beam will detect the droplet, and after the laser beam detects the droplet, it will be allotted a charge. Why? Because this charge allotment would be dependent on this fluorescence detection. So let us say you have two antibodies, one which is fluorescing red, other one is fluorescing green. So if it has red fluorescence, it is going to get, say, negative charge. If it has green fluorescence, it is going to get positive charge. So because this allotment of charge is dependent on the fluorescence, so the laser has to detect the cell and the fluorescent signal from the cell has to be detected, and then this charge would be allotted to it. Okay. So in this case, for instance, these are negatively charged due to detection of a single fluorescent cell, and these are positively charged due to detection of single non-fluorescent cell. Okay, so in this case, the sorting is such that you have fluorescence, you go in one population, you do not have fluorescence, you go in the other population, and that is how the charge is allotted. And this, you can set up the software before running the system, and you can feed how exactly you want to sort the cells. So once the cells are allotted some charge, what happens next? So the intensity of the fluorescence emitted by each droplet that contains a cell is monitored by the detector and displayed on the computer screen. Yes, the detector will monitor it. It will display to you that, okay, this is the level of fluorescence that you're getting over here. Now, as the computer tracks the position of each droplet, it is possible to determine when a particular droplet will arrive between the deflection plates. So the computer knows what is the speed at which the droplet is passing, when the droplet passed this particular point, what is the distance from this point to the deflection, what is the distance from this point to the deflection plates. So the computer can monitor exactly when a particular droplet that passed here will reach here. Because again, you have to keep in mind, there will be a continuous stream of droplets. So when this droplet which was detected here is somewhere over here, another droplet is here. So the computer is detecting a second, or I don't know, maybe an nth number of droplet at this point, while the first droplet which it had detected is already here. So the computer actually keeps track of each and every droplet and where it is. And as the droplet reaches these deflection plates, because they have been allotted some charge, they would be deflected accordingly, according to the charge, and you will collect them separately. So by applying a momentary charge to the deflection plates when a droplet is passing between them, it is possible to deflect the path of a particular droplet into one or another collecting vessel. So the computer now knows that, okay, there was a fluorescent cell here some time ago and I have given it a negative charge, and this cell has now reached the deflection plates. So let me give the deflection plates such a charge that these fluorescent cell which was negatively charged will be collected in this specific collector, and so it will give the plates the charge. Okay. So this allows the sorting of a population of cells into subpopulations having different profiles of surface markers. So again, let us sum it up very briefly. You have your cell culture over here. So you incubate it with a specific fluorescent antibodies. So now you have some of the cells, the violet ones, which are expressing the specific marker. So the antibodies have bound to it, and now these are fluorescent in nature. So a target cell type within a mixture of cells is first fluorescently labeled. Now you load the sample. The sample is prepared such that the suspension is dilute enough so that only one cell passes through at a time. Now as the cell is passing through here, there is a laser beam. The pathway of the laser beam is getting interrupted, and this forward scatter detector, which detects the size of the cells, will detect, okay, one cell was now passed through. And this side scatter detector, which is roughly at 90 degrees to the path of the laser beam, will detect the fluorescence. Okay. Now as it detects the fluorescence, based on the fluorescence detected, this electrode will impart it with a charge. So if it's fluorescing this color, it will impart it with positive charge in this case. And if there is no fluorescence, then it will be imparted a negative charge. So what is there over here? Cell mixture leaves the nozzle in droplets. Laser beam will strike each droplet. The forward scatter detector identifies the cell size. The side scatter detector, this would be SSC, will identify fluorescence. And the electrode will now assign a positive or negative charge depending on the fluorescence detected. And the positive charged cells will move closer to this negatively charged plane, and the negative charged cells will move closer to the positive charged plane. And now these cells will be collected in different collection tubes. So what about the condition we were talking where we are detecting multiple epitopes and we have, say, two antibodies present, and each of them are fluorescing a different color, and we want to sort these cells on the basis of two cell surface markers. So then this the software would be fed in such a fashion prior to the beginning of the experiment so that it will impart positive charge, say for example, when both the markers are present, or it will impart more positive charge. I'm sorry, no, it will see, it can impart only two types of charges, positive or negative. So it will impart positive charge when either of the markers are present. Fine, that is how it is fed. And the detector system is told that both of these have to be sorted separately. That is, cells which have only one marker have to be sorted separately from the cells which have both the markers. So this is where these deflection plates will actually come into play, because the SSC detector will detect the fluorescence, whether it is one fluorescence or two different fluorescence that is being emitted by the cell, and then the cells would be sorted. So this is where gating becomes important, because ultimately the sorting is on the basis of just a positive or a negative charge. So how exactly will you identify that, okay, if my cell population is showing up in this specific region of the cytogram, I will consider it to fall under this particular population. So for that, you do require a lot of prior knowledge and some experience with the particular type of sample that you're dealing with, and it is something that you have to optimize for your specific sample. So coming to the uses of flow cytometry and FACS, the flow cytometer has multiple applications in clinical as well as research problems. FACS can sort cells as fast as 15,000 cells per second to a very high purity of over 98%, followed by culturing of those cells as a pure population. So its efficiency is very, very high, and the populations are so pure that you can culture these cells without doubt of any co-contamination by a different cell population. By treating appropriately processed samples with a fluorescently labeled antibody and performing flow cytometric analysis, one can obtain the following information: how many cells express the target antigen as an absolute number and also as a percentage of cells passing the beam. Because see, the total number of cells passing the beam could be detected by the first detector or the forward scatter detector. So you can get an idea of the number of cells that are expressing a target antigen as both an absolute number as well as a percentage or fraction of the total number of cells, the distribution of cells in a sample population according to antigen densities as determined by fluorescence intensity. So, how many antigen molecules are expressed on the surface of the cell will determine how many antibodies will bind to it, and which in turn will determine the intensity of the fluorescence. So, you can even identify or detect the distribution of cells according to the antigen densities. It is therefore possible to obtain a measure of the distribution of antigen density within the population of cells that possess the antigen. Third, the size of cells. So the forward scatter detector actually detects the size of the cell. So this information is derived from analysis of the light scattering properties of the members of the cell population under examination. See, a bigger cell will scatter the light more. So how much of the laser beam is being interrupted, or what is the how much of the laser beam that was initially being detected by the first detector is being interrupted is a measure of the size of these cells. Flow cytometry also makes it possible to analyze cell populations that have been labeled with 2 or 3 different fluorescent antibodies, and this is something we have already discussed. For example, if a blood sample is reacted with a fluorescent tagged antibody specific for T cells, and also with a phycoerythrin tagged antibody specific for B cells. Now, fluorescein and phycoerythrin are fluorophores that we have already covered in one of our previous lectures. So if you are new to this channel, then do go back to the lecture on immunofluorescence to know more about these specific fluorophores. So let us say you have fluorescein tagged T cells and phycoerythrin tagged B cells. So the percentage of B and T cells may be determined simultaneously with a single analysis. And numerous variations of two-color analysis are routinely carried out, and even three-color experiments are quite common. So now a little bit about some of the difficulties that are faced when performing flow cytometry. So FACS separations are more difficult to conduct on cells from animal tissues, which interact with adjacent cells and are surrounded by an extracellular matrix, because these cells will have a tendency to clump together. Such samples must be treated with proteases to degrade the ECM proteins that attach the cells in the tissues to one another. And this protease treatment will help in the disaggregation of the cells. These proteases may also degrade the distinctive cell surface marker proteins that distinguish one cell type from another, and this is one of the detrimental effects or side effects associated with treatment with these specific proteases. Cells recovered from solid tumors also show a tendency to clump together, and should therefore be filtered through a sterile gauge, which will remove any clumps that are present. Because, as we have already discussed from the very beginning, it is absolutely imperative that you have single cell suspension, and that at a time only a single cell passes through the pathway of the laser. So if you are interested in learning more about flow cytometry, then this is one specific website that you can visit. You can just copy this link and paste it on your address bar of your web browser, and it will take you to the link and you can find out more. So to summarize, we talked about a very common technique, flow cytometry. What are the various components of flow cytometry? What is the underlying principle and how exactly it works? We then talked about an extension of flow cytometry, that is FACS, which is fluorescence assisted cell sorting, where how using flow cytometry you can sort a specific population or subpopulation of cells from a mixture of cells. So that brings us to the end of this particular discussion. I have tried my best to convey the topic to you, whatever I know about it. And in case you have any questions, any doubts, please feel free to leave it in the comment section and I will definitely get back to you. So this is the third unit or the third lecture under this unit of immunofluorescence and how basically the different detection methods that are reliant on fluorescence conjugated antibodies. So if you like this video, then do like it. And if you want to learn about more such different techniques that you use in laboratories, then do subscribe to my channel and I will get back to you next time. Bye.